morning. Thank you all for having us. So we all represent, we are part of a consortium known as the Alliance for Pathogen Surveillance Innovations, or APSI India. It's funded by the Rockefeller Foundation. Since 2021, we've been working on surveillance. It all started with surveillance of clinical samples of SARS-CoV-2 variants circulating during the second wave of COVID in 2021. We did extensive genomic surveillance. We are a multi-city pan-India consortium with our nodal headquarters in CCMB, or the Center for Cellular and Molecular Biology, Hyderabad, and we have partner institutions in Bangalore, Pune, and Delhi. And over the past few years, we have expanded our surveillance towards pathogens of other public health concerns, such as bacteria contributing to antimicrobial resistance, and besides SARS-CoV-2, other viruses such as influenza, chikungunya, Zika. And today, our topic would be a little different from what the rest of the talks are. We are talking about the role of environmental surveillance. And we'll be specifically talking about wastewater surveillance and its importance in serving as a powerful tool to predict disease outbreaks. And I would very quickly introduce our panelists here. Dr. Rakesh Mishra, he's the current director of the Tata Institute of Genetics and Society in Bangalore. His research interests are comparative genomics of non-coding DNA in the context of evolution of complexity, genetic basis of anterior-posterior body axis formation on animals, and role of epigenetic regulation in development. And he is focused on understanding the evolution of rare genetic diseases in India. We have Dr. Vinay K. Nandicoori, who's the current director of CCMB Hyderabad, and his research is focused on understanding the pathogenesis of Mycobacterium tuberculosis, which is a pathogen of public health concern in India, and is attributed a lot to MDR and XDR TB. So that the other panel members are Shivranjani Moharir. She is currently at the Tata Institute of Genetics and Society and works on environmental surveillance of pathogens, including SARS-CoV-2 and bacteria which contribute to antimicrobial resistance. And finally, we have Dr. Mahesh Dharne. He's a senior principal scientist at CSIR National Chemical Laboratory in Pune. His group works on environmental microbiology like microbiome, virome, AMR dynamics, using various tools like next-generation sequencing and culture-based approaches under the Rockefeller Foundation APSI project. So our goal is to basically talk about the role of environmental surveillance and how it can be a predictor of various disease outbreak, including pathogens which contribute to AMR. And I'm Aruna Panda. I'm the program director of RF-APSI, and my research is mostly focused on understanding the molecular pathogenesis of paramyxoviruses, and through this understanding, focused on vaccine development. So welcome all of you, and this is a plenary session. So what we'll do is we'll start with talking a little bit about our program, followed by specific talks focused on AMR and detection of viral agents in wastewaters. Thank you. I would now request Dr. Rakesh Mishra and Dr. Nandicoori to give a brief overview of what we do as a consortium towards environmental surveillance. Thank you.
Okay. Thank you. Thank you, Aruna, for the very nice introduction. And thanks for the G-SPARC 2024 for inviting us to interact with the clinician, which is so important for the healthcare and the public health in general. I would just like to say how we got together as a team to work on environmental surveillance, and this began during the mid of the COVID pandemic, and Rockefeller offered us a generous funding to do a multi-city cluster surveillance of the SARS-CoV-2. So we got together Hyderabad, Bangalore, Pune, and Delhi as a 4 multi-city cluster. And our aim was generally to look at the genomics, do the genomic surveillance of clinical samples, mainly retrospective samples and immediate cases, to understand how the variants are emerging and if there is a correlation of the new mutations and clinical symptoms or immune escape, and those are kind of aspects. So we sequenced several tens of thousands, more than 40,000 samples, and we got some understanding of how these things are happening. So then that is really established the genomic surveillance confidence, and also that we can do it and make sense out of what data is being generated. And we also began environmental surveillance at the same time, but at a smaller activity of the program, which was largely wastewater surveillance from the few cities. Mainly Pune and Hyderabad at that time. But as things went on, pandemic became a bit under control. Rockefeller came with more generous support that let's do it for 3 years, and set up the environmental surveillance, and because that is then the of use in the future. And that part has led us to reorganize our activities, and now we are doing really extensive environmental surveillance, as is wastewater waste surveillance in Hyderabad, in Bengaluru, in Pune, and in Delhi capital region around. And this has established several things now, that wastewater is a tremendous source. It allows us to see the invisible, and then we can then predict what is happening in future once we know in a longitudinal study how the pathogen is behaving. And in fact, for the country like India, for anywhere for that matter, instead of going to individual accessing samples, if we just collect the wastewater, we can even tell in the cities. And for example, Bangalore Municipal Corporation gives us the samples from their own STPs, and then we give them the result every week, and they know which place what needs to be done, at least for the pathogen that we can see. And in general, if you know your enemy, you can target and have a strategy against the enemy. Pathogens are not visible. They are visible only when they have invaded and made a problem for us. And many times, even more dangerous, many of the pathogens are largely asymptomatic. So we don't even know that they are around us, even in the people. But if you are doing the surveillance, then you have a very good idea about, for large number of pathogens, how they are increasing in the number. That means more people are infected, whether they are causing any symptom or not, because at some time, some variant can create this trouble. So I think over now past 3 years of this study, in the country, we have been able to establish, and all over the world now it's a general understanding that wastewater surveillance should be the integral part of the civic system, and it's very much affordable. Technology is there, and there is no reason why we shouldn't use the technology and take care of the problem, have better strategy to deal with the current infections and future infections. There are many, many utilities and advantages of having this system in every small and big cities. There are challenges, there are issues that need to be addressed, but this kind of conference really is a big stimulus for such thing, because clinicians who are the main factors in this, they should understand and support and look for the data that comes from surveillance, because then they can be prepared in their hospital. And of course the civic bodies that manage the city should have strategies based on this kind of output, and it should become integral part. So I'll stop at that, and I'll ask Vinay to add to this, and then we can have more specific questions. Thank you.
Rakesh has already kind of explained how the wastewater surveillance can actually give us ideas and inputs on what is the kind of prevalent bacteria and the resistance that is there is, and the viruses that are there in the wastewater. But as an application, as you go forward, you can actually make it more targeted, and kind of looking at wastewater, say outside a poultry, or wastewater from an hospital, or like that. You can look at localized wastewater and then get an idea on what is prevalent in that particular small pocket, which is what in kind of he was alluding to would help in coming up with what are the antibiotics that are being prescribed and are where you may find resistance, what would be our alternatives? So it may actually give insights to what are the possible treatment options for, for a particular population in a particular area. That is where surveillance actually helps. One thing that is expected and that is kind of known is that not while wastewater is a powerful way to look at this, it may not work for every virus, it may not work for every pathogen. So we actually look at things in very targeted way in APSI. For example, dengue is something that we look at it in the context of mosquitoes larvae rather than looking at the wastewater. And that's the kind of strategies one has to develop depending on what the questions are, so that we are looking at surveillance as a holistic matter, not limited to most of it can come from wastewater, but in addition to that we can look at it holistically and develop necessary kits with the help of and make work along with the government to make sure that this becomes part and parcel of national policy. That's our hope. Thank you.
Thank you, Dr. Mishra and Dr. Nandicoori. Now we will move on to a couple of short presentations. The first one will be by Dr. Shivranjani Moharir. She'll talk about detection of antimicrobial resistant genes through wastewater surveillance.
Tell you in brief what I am going to talk about and how it can be inferred to get an idea about the overall AMR picture in any city or any community. So what we do is we use a metagenomics based approach wherein we monitor environmental samples and then sequence them and then look at the profile of the pathogens that are there. So the way it is done is when we think of AMR, we usually think of the clinical settings as contributors to AMR, but it's not the scenario. Other sectors like poultry, animal husbandry, industry also contribute to AMR, and it's important when we are looking at the AMR picture, it's important to look at how and what is the percent of contribution of all of these sectors towards this broader problem of AMR.
Talk a little bit about environmental surveillance. So basically we are now, as Dr. Mishra mentioned, moved on to other pathogens. So we've been doing surveillance for Mpox, which has been in the news, as well as vector surveillance of dengue viruses. As you know, dengue is a big public health concern in India currently. We used to hear about malaria, now it's all about dengue. So we have got several studies talking about what are the different serotypes available in different regions of the country, from the west, Pune to Delhi. So our consortium works on several pathogens, and as far as correlating all this data to clinical presence of the disease, we work very actively with several clinicians. Some of our partners are in LV Prasad Eye Institute right here in Hyderabad, and several hospitals in Delhi, Pune. So all of the environmental surveillance data which we generate through wastewater surveillance or vector surveillance, we do work closely with clinicians to find out the disease incidence in that specific region. Whatever we are seeing in specific wastewater regions, we do correlate with clinicians. So now we're ready to hear more about AMR detection in wastewater. Thank you.
Thank you very much. So I'm going to give a flavor of how metagenomics or genomics approach can be used to get an idea about the antimicrobial resistance picture in a community. So just to give a brief introduction about what is wastewater surveillance. So wastewater surveillance had been traditionally done for multiple pathogens, including polio. It's still done. But it came into limelight during COVID-19 when a Chinese group for the first time detected the traces of SARS-CoV in several clinical specimens, which included stool samples as well. Then immediately after that, an Australian group for the first time, this Australian group for the first time showed that SARS-CoV can be present in untreated wastewater in Australia, and this study acted as proof of concept for wastewater surveillance for COVID at a community level. Then after that, it became well accepted that both symptomatic as well as asymptomatic individuals shed the viral fragments in their feces, which find the way into the environment or the sewage treatment plant. And if we are able to procure this sample, process it, extract nucleic acids from that, perform RT-PCR, one can actually estimate SARS-CoV RNA load in the wastewater, and based on that, the disease prevalence in a population can be estimated. And also, if we are able to sequence the samples, we can actually track the variants that are mongering in the community. Now, similar kind of approach can be used to understand the antimicrobial resistance picture in the community as well. So this antimicrobial resistance is carried in the form of ARGs, which are basically genomic signatures. So what is done is, we collect the samples and look for these ARGs. Now this slide shows how wastewater surveillance can be used as a complementary surveillance system to clinical surveillance. So in clinical surveillance, what happens is you collect samples from individuals who are infected. These samples are tested, and then based on the result, you know whether the individual is infected or not. In wastewater surveillance, we collect one community sample which has the infection mongering in the community, and based on the load of that pathogen, one can actually estimate how is the disease prevalence at the community level, or for that matter, how is the ARG load at community level. Now, if we look for multiple such signatures using metagenomics approach, one can actually look for either multiple pathogens or multiple ARGs in the sample. So, but before that, why study AMR? So AMR is a global concern, and like I said, it's not just the clinical sector that contributes to AMR. Other sectors like aquaculture, agriculture, food industry also contribute to AMR. And as per WHO, AMR is the top 10 public health threat to humanity. And there was a very nice study published in 2022, which estimated that in the year 2019, around 4.95 million deaths were attributed to AMR. And of that, around 1.27 million deaths were specifically attributed to AMR. And among that as well, around 70 to 80%, that is 0.93 million deaths were specifically caused by the escape pathogens that are listed here. And an annual death toll of around 10 million will be attributed to AMR if appropriate measures are not taken by 2050. And India could contribute to 20% of this death toll if appropriate measures are not taken. It's not just the healthcare sector, even the economy would be affected. There would be 7.5% decline in livestock, and the world could lose over 3.8% of its GDP if appropriate measures are not taken. So it's imperative that we understand the problem of AMR and know how to mitigate it. So towards this, I'm going to talk about the efforts of our consortium, IFCAI consortium, towards understanding the AMR scenario in 3 cities in India. The first story is about Hyderabad, wherein we monitored 17 open drains that receive influx from multiple sectors, and then collected the samples, sequenced the samples using Illumina NovaSeq sequencing platform, which is a shotgun sequencing platform. And then after doing appropriate QC, we looked at the antimicrobial resistance genes using bioinformatics pipeline. So we looked for 4 different parameters. One, how is the ARG load? Second, what are the pathogens that have these antimicrobial resistance genes? Third, what are the drug classes that have conferred resistance? And fourth, what is the resistance mechanism that is used by these pathogens? So coming to the first question, so this plot basically shows the ARGs that we see in the 17 locations in the city of Hyderabad. And what we see is 23S rRNA with mutation conferring resistance to macrolide antibiotics appears to be the most prevalent across the 17 locations that we looked for, followed by these top 5. So out of the top 5, the top 4 belong to rRNA genes, which gives us information about the mechanistic details which can be used for designing future antibiotics. So the next obvious question is, which drug class has gained maximum resistance based on the ARG profile? And it appears that macrolides have gained maximum resistance, followed by aminoglycoside, then tetracycline, then lincosamide, and 5th is alphamycin. So these are carried by the pathogens. So what are the resistant pathogens that carry these ARGs? And it appears that the pathogen profile is very diverse, which means that multiple pathogens probably carry multiple ARGs which confer multi-drug resistance. And these are the top 5 pathogens that we see in the data. And the mechanism appears to be through antibiotic target alteration, which is mainly adopted by these pathogens based on the data. Now, the WHO has this list of AMR critical pathogens, which is divided into 3 subgroups. We looked for these pathogens and the antibiotics to which they are resistant in our data. And we see that this is the data. E. coli has gained resistance to around 30 or 35 drugs either in single or in combination. Similarly, Pseudomonas has gained resistance to around 18 drug classes either in single or in combination. So it appears that the problem that most of these pathogens have gained multi-drug resistance. And not only that, E. coli has gained resistance to even the third generation of antibiotics, because we see resistance to carbapenem as well in the, based on the data. Now, we did a small exercise where we plotted where we concised the data to see how these locations behave with respect to each other, and we see that most of the locations except for 2 cluster together, suggesting that they have similar patterns across different locations, which was expected because the nature of these locations was more or less similar. And this map shows the coordinates of the sampling locations and the number of hospitals in the 2-kilometer radius of those sampling locations. And it appears that there is positive correlation with the number of hospitals in the 2-kilometer radius and the antimicrobial resistance load that we see based on the data. Now, this was for Hyderabad. Like I said earlier, we have parallel research going on in multiple cities. So this is a data from Pune. It differs little bit in the methodology, wherein the Pune team utilizes the long-read sequencing, that is nanopore-based sequencing. And what they do is they collect samples from 10 STPs from the city and then look at the AMR profile. And this is how it looks. It appears that the multi-drug resistance, which appears in yellow color, is the most prevalent one. And based on the data, that it appears that winter season has higher ARG load as compared to summer and monsoon, which is seen at total antibiotic profile as well as when you look at individual antibiotics as well. Now, this is slide from Bangalore city, wherein we have looked at the load of AMR genes in the inlet of STPs and outlet of STPs, and we could detect around 273 ARGs in the samples, and it appears that post-treatment, the load of ARGs decreases. In the fourth study, we have tried to optimize the protocol in such a way that even the, usually the pathogens carry these ARGs on plasmids or on mobile genetic elements. So this protocol basically enriches low molecular weight DNA, and that's how one can actually look at plasmid and mobile genetic elements. So now this was with respect to the data in India that we have worked on. How is the global AMR scenario? There was a very nice study published in Nature Communications wherein this particular group, sewage surveillance group, had monitored 70 locations or 70 countries and then looked at the AMR load in these locations. And it appears that macrolides, which is brown colored, is the most prevalent one in this data. The same team did a 3-year-long study where they looked at the AMR profile for 3 years. This time they increased the number of countries to 101, and this is how it appears. So this particular study has shown that how a consortium effort can help in understanding a bigger problem of AMR, and how data analysis and sample processing at one center can help in reducing the resources and can help in understanding the broader picture. So even the European Commission has proposed the implementation of sewage surveillance for understanding the AMR problem in their wastewater treatment program. And US CDC has also adopted wastewater surveillance for understanding the problem of AMR. And we aim towards implementing this kind of surveillance program in India as well. So before I hand over to my colleague who would talk in detail about the pathogen profile, I would just like to summarize that AMR is a multifaceted problem, and it is important to implement a comprehensive surveillance approach to understand it and possibly to mitigate it. Thank you.
Thank you, Dr. Moharir, for that very insightful talk regarding detection of antimicrobial resistant genes both in open drains and sewage treatment plants. I would now like to request Dr. Mahesh Dharne to talk about detection of viral pathogens in wastewater.
Spending their valuable time in understanding AMR, which is a global concern. So as spoken by Dr. Shivranjani, she's spoken about how environment can serve as a proxy for detection of AMR and how it will help and assist clinicians to take a call on rising AMR in the city. So you need not to focus on individual patient. So it's a kind of community approach. Having said that, there is another darker side to this AMR, which we observed during COVID. So I'll be talking on another side, like every coin has two sides. One side is of AMR, and another side is darker side, which is contributing to the AMR as a trigger. What we saw during COVID is that there were, the pathogen came with a very lot of surprises to everyone, to the entire globe, and it was kind of initial collapse that there were diagnostic kits were lacking and many other things. We were not able to understand the landscape of this new variant. So what the title itself says that wastewater surveillance of SARS-CoV as an early warning system and beyond. So this is old science. There is nothing rocket science in this. Wastewater surveillance has been used in the past for polio surveillance. It's being used today also, and also for rotavirus and other enteric viruses. So what we thought in the beginning of this pandemic, whether this technology or this science can be useful in a country like India and other low to middle income countries to understand landscape of COVID. So what is it? As I mentioned, it's a old science, and it basically gives you community-based approach where you can detect the pathogen very early ahead of time. As in COVID, we know that most of the patients were asymptomatic, and they were kind of acting as a super spreaders in the community. And so there was no way to understand how this is spreading and all. So first study came from Spain, that effectively they could detect SARS-CoV and correlate with the clinical cases. So not with our surprise, in India we geared up our socks, and fortunately our leadership is here, Dr. Rakesh Mishra, who was former director of CCMB in Hyderabad, this city, and Dr. Vinay Nandicoori, who is now heading. So they look at the visionary scope of this science when it comes to country like India or Southeast Asia, how it can be used. So we all 3-4 labs came together as a 4-city surveillance project. Initially, so as I said, slide was little delayed, that COVID and AMR, these are the partners in new crime. Crime is already there, but this is a new crime. So you need to understand both, how AMR is progressing and how it is related to COVID. You know, lot of antibiotics, antivirals were used. And in India, we saw a pandemic within pandemic, that is mucormycosis. Fortunately, it was controlled at the right moment. Otherwise, it would have been a really a disaster. So as I come from National Chemical Laboratory, Pune, I will take that slide later on. First, I will go into the first data came from Hyderabad during this COVID pandemic. The gentleman you see in the center is right away sitting on the dais here as a panel discussion member. And this study was done in collaboration with CCMB and IICT, where they put forth and standardized a lot of various protocols and to make the SOP, because this was never done in India outside the scope of polio. So quite a little challenge. And Dr. Venkat Mohan and Dr. Shivranjani and Dr. Mishra's team came together, and they put forth a proof of concept, say in around August 2020. And things were quite better for us to adapt in other labs in India, whether it is Pune or Bangalore or Delhi or now in Shillong also. So this was very successful proof of concept which we adopted, and this study is from Bangalore, where Dr. Farah Ishtiaq is looking after. And so in Bangalore, from 25 STPs, samples are still being collected. This work is, by the way, not yet stopped. This is still going on right from the day we started. And today also we can tell which variant is prevalent in these cities. So this is very active surveillance going on, although we know that clinical surveillance is kind of stopped because patients are not coming for regular testing, and there is a lag. So we'll be still continuing this surveillance for a few months, as we know that winters in last 3 years have always gave us a lot of surprises in terms of COVID variant, whether it's Omicron, XBB, JN1 or very latest KP, and now the lot of progenies have been there, and now the current variant is XEK, which is combination of XCC and KP variant. So a lot of things are happening, and environmentally serves as a sink for detecting all these. And Bangalore study has been published in Lancet Regional Health last year. However, the aim of our efforts was not to publish or publicize our portfolio in terms of CV, but how this work can be very useful in a country like India as a policy making. Like previous 2-3 presentations, we saw the focus was given on the toilets. Sir was telling that why better cleaning of the toilets and all, because that also serves as a reservoir for AMR and other things. So I will tell you in the forthcoming slides how we used hospital wastewater to track COVID. That also I will touch upon. So after Bengaluru, I will enter in the Pune, where first study we found Delta variant in the Pune STPs. This study was sponsored by Science Engineering Research Board, ACRB DST. This was the first project, was small although, sponsored by DST to support this small work in Pune, thanks to CSIR and ACRB, which helped us to build confidence that yes, the technology works. So this is how we detected, that time it was called as double mutant earlier, when it was not designated as Delta. So this study this was very helpful for our policymakers who are sitting in the health department in local municipal corporation to know what regulations or what control they have to do in different wards of the city. So this small study that gave us a lot of confidence. Then partners started coming, like CCMB, TIGS, Ashoka University, IGIB New Delhi, NCL Pune. A lot of people came together and we built a consortium which is called as APSI, Alliance for Pathogen Surveillance in India initiative. So under APSI umbrella, we multiple labs indicated here are working. And very importantly, we got support from local municipality authority and health departments, that is Pune Municipal Corporation and Pimpri-Chinchwad. So we have twin corporations in Pune. So with their support, you can see on right hand side, we have reported till now Delta, Omicron, XBB, BA2.86, JN1 and KP variants in wastewater before they started appearing clinical cases. So now gears were switched. So health authorities were asking us what is the trend of SARS-CoV in wastewater. So see, this is how, yeah, okay, sure. Yeah, I will, so almost done. So this is the standard work protocol we follow in the lab. And every week and every twice a week, data goes to municipal authorities so that they can be well informed to take a policy decision. This is what the data I will rush through, that we reported Delta, Omicron and other variants in the wastewater. The interesting part of wastewater surveillance is it can detect the silent waves also. You can see here that cases were not there, but still the STP samples had viral load. So what does it mean? So either vaccination gave protection or natural infections give protection, but in wastewater still you see the viral load. And coming to that later, we also reported JN1 variant in Pune in last year. And so here in this study, we also used hospital wastewater. You can see here, this is government hospital in Pune. We took samples from 4 wards and we could detect there also JN1 variant. So it's not only domestic municipal STP, but also hospital wastewater can also serve as a early indicator. Then eventually we reported KP signals also in the wastewater. So here you can see the overall trajectory of how different variants were reported from Pune. And also, as I mentioned, our efforts are still continued, and we have gone beyond COVID now. Along with COVID, we are able to successfully track influenza, different influenza A, namely H1N1, H3N2, and also RSV, well ahead early in the wastewater. So looks like the technology is promising and it's working. So what next? So our effort have been, so we designed the dashboard, especially for Pune city, and it was caught by WHO environmental surveillance portfolio, And here on right-hand side, you can see that it has been placed on the WHO website as well. So anybody sitting in the world, anywhere in the world can track what is the status of SARS-CoV and other H1N1 viruses in the wastewater samples. So this is how, just like clinical dashboards like what ICMR and NCDC has. So similarly, wastewater surveillance dashboards also are very useful in a country like, diverse country like India, whether AMR is uniform in the country or it is geographically specific AMRs are present in country. That's why these dashboards are very useful. So this is impact of our APSI network. And this is the last slide. So what are our goals? Whether wastewater surveillance can become a policy in the hospitals and also under different sectors under One Health. Now we know that AMR is not restricted to a particular sector. It has been in environment, wildlife, hospitals, aqua, and so many. Can we predict and track long COVID situation? Because this is another side effect of COVID. And also very futuristically, whether AI/ML based predictions can work based on environmental surveillance in low to middle income countries. As our colleagues mentioned, we are also gearing up for surveillance of monkeypox using wastewater or environment as a source. So thanks to CSIR, APSI, and thanks to the G-SPARC 24 organizers to give us opportunity to speak our contributions in environmental surveillance. And as I mentioned, this is a need of ours, so we need to unite and work together, whether it is a hospital sector or different AMR One Health sectors like aquaculture, poultry, and so on. So with this, thank you very much for giving us opportunity to present our efforts. Thank you.
Thank you so much, Dr. Dharne, for that very insightful talk again, how to detect pathogens of concern in wastewater. We would now have a little bit of a discussion where the questions are also open to the public. If you have any questions, feel free to ask our expert panel here. I would start with a general question to Dr. Mishra. As you know, wastewater-based epidemiology is picking up in several countries, including the Southeast Asian nations, the United States, and even many European countries are taking it very seriously on how to use wastewater-based epidemiology to predict disease outbreaks, since it can be used as a forewarning before the outbreak actually happens. In terms of India, what are your thoughts on utilizing this technology which APSI and other consortiums have developed to have a sustainable surveillance program to detect diseases in India?
Thank you, Aruna. So that is really the important thing. This, what you heard, that lot of deep science has gone in. We have generated protocols which can be used for this. So now, but doing in 4 cities is not enough. It has to be done by a whole country. So we have been having a series of talks with the top leadership in the country in different departments. And NCDC has really come forward with a very positive way. And we are now sharing the protocols with them. And at the same time, we have been interacting with the industry partners to make the kits which are totally indigenous and made available for the surveillance purposes at a very minimal cost. So those are the 2 things that are required. So implementing agency and supplies of material, and we provide the SOPs and handhold them for sometimes certain training and whatever data analysis or any other help is required. And I see that whoever, and the other stakeholders are the local city, and we get lots of requests and interest from cities, particularly in Bangalore, have been so cooperative, and in fact they have been using our data to go more granular where the cases are and how you can do. You don't have to close the whole city if the infection are increasing in one region of the city and so on. And the trust we have generated by doing this is something which makes us feel more motivated and more involved and engaged with the government agencies, because they have to take further this whole process. And I'm sure in coming months, including you may have recently heard Niti Aayog come up with very broad agenda of pandemic preparedness and emergency response, and surveillance is going to be the really basic factor in that. I can just mention very briefly, because the trust that we have generated when Mpox came and I mean the news came and WHO declared it as a problem. Karnataka government approached us that can we do surveillance from the airport samples of Bangalore and Bengaluru, and that was very nice. Of course, we can the NESA we can develop in matter of few weeks, because it's just standardizing the primer pairs, because these are all very standard technologies. As long as you are doing nucleic acid-based surveillance or diagnostics. So we are now monitoring the airport every day, arrival of international flights, and so that is very good things. It makes us feel happy that the cutting-edge technology is now being made available, and there are takers who are using this technology, and I'm sure its consequences will be better public health and healthcare, and in a larger context of One Health, we have to keep doing this. Yeah.
Thank you, Dr. Mishra. The other point I wanted to emphasize was for Dr. Nandicoori. As you know, Mycobacterium tuberculosis is a huge problem and we are having all these multi-drug resistant and extremely drug resistant strains. What are your thoughts of using environmental surveillance for pathogens of such importance?
Mycobacterium tuberculosis, which causes TB, is actually a very, very slow growing pathogen. To put things in perspective, a E. coli, which is pretty much like a bacteria, divides every 20-25 minutes, whereas Mycobacterium tuberculosis takes approximately 20 to 24 hours. Second thing is it resides in your host immune cells. So the load in the wastewater is likely to be lower, not because the infection load is less, but one, because of its time it takes to divide. Second, the niche area is different. Given this, you may not find much of Mycobacterium tuberculosis in the wastewater. However, that's what I was telling a while ago, every pathogen requires a specific and a proper way to do surveillance. Like if you are going to do dengue, you have to do with the mosquito larvae, and if you are going to do with Mycobacterium tuberculosis, you will have to do with the wherever the infection is from as much as possible. Second aspect is the multi-drug resistance. At this point of time, even today, Mycobacterium tuberculosis kills 15 lakh people per year. To put things in perspective, SARS-CoV-2 in its whole pandemic has killed approximately 80 lakh people. So 15 lakh per year means in about 5 years, it will cover as much as SARS-CoV-2 pandemic. And that's continues to be a major threat. And one of the reasons it's a major threat is because of multi-drug resistant pathogen arrival. Now 15 to 20% of the cases are actually multi-drug resistant, and the mutations that cause resistances are not completely known. So as part of a Department of Biotechnology initiative with ICMR, we are actually working on sequencing 30,000 clinical tuberculosis genomes to identify what kind of mutations in India that are found in multi-drug resistant pathogen, that is tuberculosis, and make that a part of future surveillance strategy to find out when a patient walks into the clinic, what is the possibility of what resistance they may have, and accordingly decide the what kind of drugs can be given. So this is the information we can provide to the doctors, and that can be used as a way to actually come up with a better treatment line. So that's where we are, and I really hope, I can talk more about tuberculosis, but due to time constraints, I would leave it here. Thanks.
Thank you, Dr. Nandicoori. This wraps up our plenary session. Thanks to G-SPARC for giving us the opportunity, and I hope by the end of this session, we could convince you the importance of environmental surveillance, specifically wastewater surveillance, in predicting diseases of public health concern, including bacteria which contribute to antimicrobial resistance. Thank you all very much.